Good afternoon, class. I have just been informed that somebody in this class got very sick last week. That is why I have brought an inspector with me.
So from here on out, we're going to do individual investigations for everybody to see why Jennifer's sick. She's terribly ill, and we have to find out who's at the bottom of this. Hello. Well, you are suspect number 1. As you know, Jennifer is very ill, and I'm checking out everybody in the classroom.
Well, I would have done it to Jennifer.
Sure. What's your name?
My name's Dahydra. And I'm a curved-shaped bacteriophage. Once upon a time, I became infected by a virus that integrated its genome into mine. Now I have the ability to produce clarotoxin. If I'm consumed and make it past the acidity of the stomach, I use my flagella to breach the intestinal wall. Once inside the intestinal wall, I enter the lumen where I release the clarotoxin. The clarotoxin I release is a 6-subunit protein with an A subunit and 5 B subunits. The B subunit binds to glycosides on the intestinal epithelial cells. I am packaged into vesicles and escorted via endocytosis and then taken into the cytoplasm where my A subunit is detached from the B subunits. After separating, my A subunit is exported into the cytoplasm. My A subunit attaches onto an ADP ribose to an amino acid with the host G protein that regulates adenyl cyclase. Adenyl cyclase is continually released, activating the ion transport system. The ion transport system causes sodium, chlorine, water, and bicarbonate to exit the cell. This induces watery diarrhea and dehydration.
Hey, you're my suspect number 2. So I'm sure you know Jennifer's sick. What's your name?
Fluenza.
Fluenza. Okay. Well, you know, Fluenza, if you would have done it, how would you have went about it?
Well, if I would have done it, I probably would have sneezed on her pen, but I would apologize, of course.
Okay.
But what they wouldn't have known is that I contain little influenza viruses in my body that would attack their epithelial cells in the respiratory tract. During attachment, the virus would have hemagglutinin glycoproteins that bind to the celiac acid receptors on the surface of the host cell to initiate the infection. Binding allows the uptake of the virion through a process of receptor-mediated endocytosis, which would engulf the virus by the host cell's plasma membrane and then internalize it in an endosome. That endosome with the envelope layers of the virion would completely disassemble, leaving just the negative single-stranded RNA genome to be released into the nucleus for transcription. Inside the nucleus, the RNA genome must first be converted to positive sense mRNA genome by the RNA polymerase to complete transcription. Then the host cellular machinery would translate viral proteins from mRNA in the cytoplasm, which would allow for further replication. With the help of my newly translated proteins, my virus could form the proper virion shape and package the RNA genome to finally be released into the host cell. My virus would make its final exit by budding off of the host cell through cleavage of the celiac acids receptors by the neuraminidase glycoprotein. But that's all hypothetical, of course.
Okay, you're my suspect 3 for today. What's your name?
My name is Paulori.
Okay, well, do you know why you're here?
No, I do not.
Okay, we're investigating you because Jennifer is very ill, and we need to figure out who did it.
Well, I don't know why you're asking me. You need to ask staff, the bully.
Well, right now we're doing you. So if you would have done it, how would you have infected Jennifer?
If I would have done it, I would have spit in her drink while she wasn't looking. My primary target is the epithelial cells, which are in the mucosal lining of the stomach. The primary target for me is the inner surface of the epithelium and occasionally inside the epithelial cells themselves. I sense the acidic pH in the mucus lining of the stomach, and I move toward the less acidic region by chemostasis. I use flagella to bury myself inside the mucus lining of the stomach to reach the epithelial cells underneath. Pylori, myself, adheres to epithelial cells by producing adhesions that bind to lipids and carbohydrates inside the epithelial cell. I neutralize the acid in the mucus by producing large amounts of urease, which breaks down urea in the stomach to carbon dioxide and pneumonia. I use vacuolating cytotoxin that damage epithelial cells by disrupting tight junctions and causing apoptosis. I also use cytotoxin-associated gene A that causes inflammation by inhibiting apoptosis and triggering the whole cell growth. But that's hypothetical, of course.
Man, it's about 12:30. I'm ready to get out of here. All right, so you're suspect number 4. Let's wrap this up. What's your name?
Berkeley?
Berkeley. Okay, Berkeley. Well, do you know why you're here?
No.
Okay, well, Jennifer's infected, and she's very ill. We got to get to the bottom of this.
Oh, no.
Yeah, so, hello?
I'm sorry.
Okay, so, if you would have done it, how would you have did it?
Well, I am spread by droplet transmission via the air when a contaminated patient coughs or sneezes. I am a large non-motile rod-shaped obligate anaerobe, a facultative intracellular parasite usually made of macrophages. The major virulence factor is located on the cell wall structure of Mycobacterium tuberculosis. The lipids in its cell wall contain mycolic acid, which is a unique alpha branch lipid that affects the permeability of the cell's surface, preventing attack from lysosome, cationic proteins, and complement deposits in serum. Also, the bacteria has special mechanisms for cell entry, binding directly with mannose receptors on macrophages via a particular glycolipid or indirectly through complement receptors.
Okay, last suspect of the day. What's your name?
Staff?
Okay, so do you know what's going on?
Yeah, someone's getting sick in class, right, Jennifer?
Right, it's
Yeah.
Jennifer. So you already know. So I'm going to ask you a few questions today, okay?
All right.
So do you know who Pylori is?
Yeah, she likes to instigate stuff in class.
Okay. Well, I didn't really ask you that, but okay. So do you know who Influenza is?
Yeah. She likes to suck up a lot.
Okay. All right, so if you would have infected Jennifer, how would you have done it?
Well, we take a gym class together, so probably would have done something in gym, like the locker room, because I get transmitted through bodily fluids, like sweat and stuff like that.
Sounds dirty.
The leftover bacteria in the area can enter the body through cuts, anything like that. I attack the macrophages of the cells. When white blood cells go around to collect the bacteria and capture it, I can change the way that they're formed. I change them into a 2-deoxyadenosine, which allows the macrophages to be not really involved, if you know what I mean. They don't help the white blood cells collect anything. So on top of that, I can delay my infection for up to 2 days. So that would further hide that it was me. That's about it, though.
Darren, put your phone down, please.
Hey, sorry to interrupt your class, Mr. Patel.
It's okay.
After 5 hours of investigation, we finally figured out who did it. I thought it was one of you guys, since most of you guys infect epithelial cells, but it's not. Actually, it's your teacher over here. Come to find out, Jennifer was infected by the bacterium Listeria monocytogenes, aka Mr. Patel over here. Mr. Patel is the bacterium that converts into an intracellular pathogen from a saprophyte. The bacterium internalizes in the epithelial cells of the GI tract. Internalization may be caused by phagocytosis or induced phagocytosis. Listeria uses the internalin A and internalin B cell wall adhesion proteins to bind to cadherin and MET proteins in the host cell membrane. When the phagocytic vacuole enters the cell's cytoplasm, Listeria lysine O, which is a phagolysosome produced by the bacteria, lyses the vacuole and allows it into the host cytoplasm to multiply. In the cytoplasm, Listeria spreads between cells through actin-based motility. Actin is formed by the bacterium's surface protein ACT-A. When mobilizing, the bacterium moves to the cell membrane and creates phylopods, which are extended parts of the cell membrane that includes the bacterial cell and reaches to adjacent cells. Then, the adjacent cells ingest the phylopods, and the bacterium enters the cytosol of the new cell. As you guys know, this type of crime is punishable by law, so don't you guys even think about it. You, Mr. Patel, is going right downtown.
No, I'm not. I refuse to go.
Yes. Oh, yes.
No. Don't you dare. Don't.
Yes, you are. Other hand, sir. Come on with me. Y'all have a nice afternoon.
I'm innocent. I'm innocent.